Today I'm going to present my research proposal on the topic Development and Therapeutic Potential of Folic Acid Coated Iron Oxide Nanonutraceutical, an integrated approach to address anemia. These are the points which will be discussed throughout the presentation, starting with the introduction of anemia, its prevalence, current treatments, and their limitations, to understand the need for formulating the nanonutraceutical, the objectives of my research, the methodology that would be adopted to conduct my research, and the results that I expect from my research. Starting with the introduction of anemia. It is a Greek word that means lack of blood. It is defined as decrease in the number of red blood cells or the amount of hemoglobin in the blood. Hemoglobin is a protein which is present in red blood cells. Its main function is to carry oxygen to various cells for the process of cellular respiration in order to provide us energy. So anemia may lead to the state of hypoxia, which means lack of oxygen. Anemia can result from various underlying causes. For example, deficiency of nutrients like iron and B vitamins, chronic diseases like chronic kidney disease and inflammatory bowel disease, blood loss that might be due to some injury or during the process of giving birth, and infections such as malaria. Symptoms of anemia are related to the reduced oxygen carrying capacity of the blood, causing fatigue and shortness of breath. During this condition, the heart rate increases to pump a larger volume of blood to compensate for the reduced oxygen carrying capacity. Reduced supply of oxygen to the brain cells may lead to headache and dizziness. For the treatment of anemia, we may consider dietary modifications, incorporating food sources rich in iron and B vitamins like folic acid, micronutrient supplementation, and blood transfusion when hemoglobin levels falls below 6 gram per deciliter. Over here, the laboratory tests are enlisted for the diagnosis of anemia, like complete blood count, that includes HB levels, RBC count, mean corpuscular volume, and hematocrit levels. Serum iron levels accounting for the amount of iron circulating in the blood. Ferritin is a protein that stores iron in cells, especially in the liver. Total iron binding capacity measures the maximum amount of iron that can be bound to transferrin, a protein that transports iron in the blood. According to the recent statistical report of World Health Organization, 24.8% of the global population is suffering from anemia, out of which 35% are pregnant women, 28% are the non-pregnant women of the childbearing age, and 37% are the children in the age span of 1 to 5 years. In 2018, National Nutrition Survey was conducted by UNICEF. According to its report, 45% population is suffering from iron deficiency anemia. Out of 23% are the pregnant women, 19% are the non-pregnant women of the childbearing age, and 58% are the children in the age span of 1 to 5 years. Various synthetic salts of iron are currently being used as iron supplements. They can either be administered orally or intravenously. For example, ferrous sulfate, which is the first generation of iron supplementation, ferrous gluconate and ferrous fumarate, which are known as second generation of iron supplements, are the salts which are used in oral supplements. They provide 60 to 80 mg of iron per day, but they have few limitations. They are less bioavailable as they may combine with food components such as phytates. They may also cause gastrointestinal discomfort, causing nausea, abdominal pain, and bloating. On the other hand, iron dextrate, iron sucrose, and ferric carboxymaltose are the salts which are administered intravenously. They provide 80 to 160 mg of iron, but the rapid delivery of iron directly into the bloodstream may cause iron overload in the tissues and organs, causing oxidative stress. They may also cause iron intoxication, leading to symptoms such as nausea, vomiting, and abdominal pain. So there is a need to move to other salts of iron which are more bioavailable, stable, and shows less adverse effects. Nutraceuticals is derived from the combination of nutrition and pharmaceutical. Nutraceuticals are typically defined as pharmaceutical-grade standardized nutrients derived from food sources that, in addition to providing basic nutrients, also provide extra health benefits. They mainly differ from pharmaceuticals in that they do not provide a pharmacologically active ingredient and instead deliver nutritional supplementation for medicinal purposes. Nanotechnology is a field of biotechnology that manipulates and handles materials at a nanoscale. Nanonutraceutical, as the name suggests, nutraceuticals at a nanoscale. They have a number of characteristics that make them superior over traditional nutraceuticals, such as enhanced bioavailability and targeted delivery due to a smaller size, and improved stability because of increased surface area and charges on their surface. The hypothesis of my research is folic acid coated iron oxide nanoparticles may improve iron bioavailability compared to conventional iron supplementation. The objectives of my research are formulation of folic acid coated iron oxide nanoparticles, then the characterization of the prepared folic acid coated iron oxide nanoparticles, and then to assess the suitability and bioavailability of nanoparticles as advanced nanonutraceuticals in biological model. Now coming to the materials and the methodology that would be used to conduct my research. Experiment number 1 is the synthesis of iron oxide nanoparticles. The first step would be the preparation of spinach leaf extract. For this purpose, spinach leaves will be collected and washed. The leaves will be air dried at room temperature and will be grounded into fine powder. Spinach leaf extract will be produced by boiling 5 grams of powdered spinach leaves in 100 ml of distilled water with continuous stirring for 20 minutes. The mixture will be cooled to room temperature and will be subjected to centrifugation at 6000 rpm for 15 minutes to get the aqueous leaf extract. Iron oxide nanoparticles will be synthesized by using the aqueous spinach leaf extract and ferric chloride in 2:1 volume ratios and will be heated at 60 degrees with continuous stirring using magnetic stirrer for 30 minutes. The green color of the spinach leaf extract of the mixture will change to blackish brown color and the precipitates of the iron oxide nanoparticles will be formed. The mixture will be allowed to cool down to room temperature and the iron oxide nanoparticles will be obtained by centrifugation. The nanoparticles will be washed thrice with saline water and will be dried at 60 degrees for 24 hours. The next step would be the coating of the prepared nanoparticles with folic acid. 0.2 gram of folic acid will be dissolved in 1 molar 40 ml ammonia and will be added to the prepared nanoparticles. For a further 30 minutes, the mixture will be stirred constantly until a precipitate will be formed. The precipitate will be separated by centrifugation and washed thrice to remove excess ammonia. Then it will be dried in oven at 60 degrees for 24 hours. For the determination of the structure, morphology, composition and stability, the enlisted tests will be used. X-ray diffraction analysis will be used to determine the crystalline structure of the synthesized iron oxide nanoparticles. Transmission electron microscopy will be used to determine the shape and size of the iron oxide nanoparticles. In order to determine the functional groups present in the prepared nanoparticles, Fourier transform infrared spectroscopy will be used, and to determine the iron content present in the nanoparticles, UV visible spectroscopy will be used. To determine the stability and particle size in a solution of the nanoparticles, zeta sizer will be used to determine zeta potential and particle size. To determine whether the particles are homogeneous or not, Pauli dispersity index will be determined by using zeta sizer. Experiment number 2 is to check the bioavailability and consequential effects of the fluorescent coated iron oxide nanoparticles. Of the study will be 12 weeks. Anemic variant will be prepared for inducing anemia in rats. The 48 rats will be divided into 8 groups, each having 6 rats. One group will act as normal, that will be the non-anemic group. In rest of the 42 rats, anemia will be induced by giving phenyldihydrazine injections at a dose of 20 mg per kilogram body weight for a period of 1 week. After 1 week, blood samples and organs will be collected to check whether anemia is induced or not and for the baseline data. The treatment design is shown in the data. Group A is the normal non-anemic group. It will be given the basal diet at 20 gram per kilogram body weight per day. Group B will act as positive control. It will receive 3 mg per kilogram ferrous sulfate, the commercially available iron supplement in the market, and the basal diet. Group C will be the control negative. No treatment will be given to this group, just the basal diet at 20 mg per kilogram body weight. From Group B to Group H, these are the 5 treatment groups. They will be receiving varying amounts of the prepared folic acid coated iron oxide nanoparticles from 1 mg per kilogram of body weight to 5 mg per kilogram of body weight along with the basal diet. The rats will be physically analyzed after every 15 days. Their feed intake and weight gain will be monitored. After every 30th day, blood samples and organs, that is liver and kidney, will be obtained to check the toxicological effects and bioavailability of iron. For the interpretation of the data obtained after physical and biological analysis, 2 days of analysis of variance and the CRT followed by 30 HSD will be performed using Statistics 8.1 software at a significance level less than 0.05. Expected outcomes of my research project are improvement in anemia management based on the formulated nanoparticles and potential advantages over conventional therapy. The references of all the literature cited in my presentation are enlisted. Thank you so much for your time and patience.